People are still joining, but let me not take any more of Richard's time by a long introduction, and just say it's a great pleasure to introduce today's Anderson Lecture distinguished speaker, Richard Henderson. I've known Richard for only about 40 years. Um, Richard did his undergraduate work in physics, University of Edinburgh, and then moved to biophysics, doing his PhD at University of Cambridge with David Blow, an eminent crystallographer, working on chymotrypsin. So Richard continued working on chymotrypsin as a postdoc at Yale, and then he came back to the Medical Research Council Laboratory of Molecular Biology, where he's been ever since, and has had a spectacular career championing and extending electron microscopy and high resolution studies using EM. So I could spend the next hour describing, many of the things Richard's done. Um, one of the most one of the earliest and most important contributions was famous work in 1975 in collaboration with Nigel Unwin, where they were able to use um, uh, electron microscopy of unstained samples. It was sugar embedded bacteria rhodopsin and visualize the alpha helices. Now, this is phenomenal for a number of reasons at the time. One is it was actually, as far as I know, the first direct visualization of secondary structure in an integral membrane protein. It had the unfortunate consequence that many people extrapolated from this one structure to conclude that all integral membrane proteins are alpha helical. That's the problem with great extrapolations from one structure. But it also showed that you could visualize secondary structure in a protein without destroying it in the electron microscope. So Richard has gone on to pioneer many things in high resolution microscopy, including playing a key role in developing direct electron detectors, which hopefully he'll talk about. And as a result of this, in 2017, he shared the Nobel Prize in Chemistry with Jacques Dubochet and Joachim Frank for their incredible contributions to the field of cryo-EM, which has now emerged as perhaps the main structural technique for solving atomic structures of macromolecular complexes. So I don't want to say any more. Richard has a tremendous amount to say. Let me just explain about questions at the end. We're going to use the chat function. So if you have a question during Richard's talk, do chat to all so other people could see it as well. And I will then select questions to ask and unmute people to allow them to ask those questions. So with that, let me turn this over to Richard.
Yeah, so thank you very, very much, Ed. Indeed, it has been 40 years. I'm so sorry I'm not visiting you in person. We've planned this for several years, but at some point I'll be there. And it'll be much more fun because you get to see what everybody's doing. But let me put my shared screen up and see if we get, you should see the title slide now. So yeah, so today I'd just like to tell you a bit about electron cryomicroscopy, and it has various forms, and then show some of the highlights that are not too old, sort of relatively recent highlights, some of the outstanding issues, and then some of our own current interests. So I quite like to start with this first slide, which comes from a review that, and apologies for those of you who are crime experts who've seen this one before, but Tim Baker and I were asked to write a chapter in the International Tables for Crystallography, which is mainly X-ray crystallographers, that was being edited by Michael Rossman and Eddie Arnold. And there were only, out of the 90 chapters, there were only 2 that dealt with electrons. One, electron diffraction, which Hua Chu wrote that chapter, and then Tim and I were supposed to write about imaging. So we thought we'd make this one figure that tried to give an overview of what is cryo-EM. And it showed state of the art round about the year 2000. And there's 4 panels that were the 4 different levels of ordering from single particles embedded in a thin film of amorphous ice without any symmetry. That was the 70S ribosome of Joachim Frank's work. Then an icosahedral single particle, hepatitis B virus cores, decorated thin filaments from muscle, from Ron Milligan's work, and then 2D crystals, which was not, and Tim and I decided we shouldn't put any of our own work in, so we picked the best work that we could find that was representative of the field. And this was a 2D crystal of the light harvesting complex from Werner Kuhlbrandt's work, which gave the second sort of near atomic resolution model of a membrane protein. But each of the panels shows where we were around about 20 years ago. And the first one came from Joachim Frank's group. He was then in Albany. Gabashvili et al. Had an 11 angstrom structure of the E. coli 70S ribosome, and they could see small subunit, large subunit, tRNA, a long bit of RNA double helix, but no real sort of high resolution details. The first sub 10 angstrom, sub nanometer structure was Bettina Botcher, who was a postdoc with Tony Crowther here in Cambridge. And that was a 7 angstrom structure with these protrusions you can see in the images. And then the model that she got is on 10 times higher magnification. You can see the protrusions have little ridges down them. That is a little 4 helix alpha helical bundle. And they were able, even though it was only 7 angstroms, because they knew a lot about the polypeptide, they could trace the chain and have a sort of pseudo atomic model, but it wasn't really good enough. And then the third panel is from, and of course Ed's worked a lot on this as well, but Ron Milligan did difference maps at 30 angstrom resolution in 1995 of actin at the middle of the fibers in blue, tropomyosin in green, and then different domains of the actin, of the myosin that was used to decorate it in rigor in this. And they were able to find out a lot about how the thin and thick filaments move even at this low resolution. Of course, all of these now are at higher resolution. And then Werner's light harvesting complex did give them an atomic model, 25 kilodalton protein in little trimers, 3 transmembrane helices, 13 or 14 chlorophyll A and Bs. And that was a really good, but you could see that it was possible to do good work, but resolution was limited. And if you were making a similar diagram now, you'd probably add a 5th or 6th panel, the main one being electron tomography, which had barely started then, but now you can get quite good resolution. And if you do sub-tomogram averaging in the same way that you do single particle averaging, you can get quite high resolution out of tomograms. And I'll show a couple of slides of that. But that's where we were. And then I think, although many people have made contributions, for example, Bob Glazer and Ken Downing froze catalase crystals back in the 70s. Joachim and Marin van Heel worked on ribosomes long before cryo existed, developing single particle work, single to noise, and so on. It was Jacques Dubochet's work that really made the big difference. And he was a young group leader at EMBL, having come from Switzerland in 1978, and with Alistair McDowell and Mark Adrian, developed this very simple method of making a thin film of amorphous water or buffer with different molecules in it. And that's their 3mm EM grid on a pair of forceps. And then Alistair McDowell was the first one to plunge freeze it into liquid ethane, which allowed it, the thin film with water, to cool very quickly so that it didn't crystallize. And then Mark Adrian was the one who developed the blotting paper that just blotted the thin film, a droplet of water, 2 or 3 microliters, down to a very thin film, and gave them a very simple, and it has a lot of advantages. You know, the filter paper removed crap off the surface. So there's a lot to be said for this. And this has all been automated now. And these pieces of equipment they developed now costs 50 or 60,000 pounds, and it has a little computer. So instead of pressing a foot switch, you do your mouse, and then it does it. And that's Jacques. So that was a very important sort of foundation of the field. And one of the first images they took was this picture, which was on the front cover of Nature in 1984, and it's the adenovirus. And you can see that most of them look beautiful, even though at that time the resolution wasn't high, but you can see a few individual subunits that have fallen off. You can see the spike, and then a couple of the virus particles have completely disintegrated, and a couple of other ones are partly destroyed. And that's probably to do with how harsh this procedure of doing the blot. And so a lot of the, and I'll come back to this a little bit later, that's part of the task for the future, is to try and develop, understand what's happening and try and find better ways around it. But that was 1984. And because computers weren't so fast and programs hadn't been written, it took actually until 1991 when Phoebe Stewart, who's now I think at Case Western, was a postdoc at EMBL and took images like this and got a 35 angstrom structure of the adenovirus in 3D. And the reason the resolution wasn't very high is that the microscope had a tungsten filament. It was a bit of a fuzzy illumination. And so the subunits, you can see them, but they're a bit fuzzy. And you don't get much more out of the images when you extract it. So the high resolution Fourier components in the image have been lost just by the technology of the time, limiting you to 35 angstroms, described by this temperature factor, B factor, fading factor of about 3,000 square angstroms. And it wasn't until about 20 years after this was done that Hong Cho, with the new generation of better microscopes, the Cryo system they had at UCLA, took similar pictures of adenovirus. And you can now see, instead of sort of one blob for the subunits, you can now see a lot of internal structure. And when they processed images like that, that's the same image top left, they got a complete structure like this on the bottom. And then by averaging some of the subunits that are present in more than one copy per asymmetric unit, you can see they got resolutions beyond the beta sheet spacing, 4.8 angstroms. So it's a 3.6 angstrom structure. So 10 times the resolution, 10 times lower B factor, 1,000 times as much data, and a real atomic model for the adenovirus. And so Jacques and the plunge freezing, and particularly with the icosahedral viruses early, showed that there was a lot of promise in the method, not only in electron crystallography. So then, what Ed mentioned before, we'd always been interested in film. And originally, when I worked with Nigel Unwin, we thought the limitation was due to the film and the film scanners. So we spent years and years investigating film and film scanners, but it never got better than about a 30% detective quantum efficiency. And that's partly because the electrons scatter when they hit the film. So we got involved with a group at Rutherford Appleton Lab, which is part of Harwell, Renato Turchetto and Nicola Guerini, and helped to develop one of the new generation of CMOS detectors. That was the Falcon detector, which is now 1234. Direct electron was Southern California, and K2 was from Catan in Northern California. And all of these detectors performed better than film, but also all of them gave you an output that could be stored as a little movie, so that if your specimen moved due to drift or radiation damage or a little bit of charging, you could then deconvolute that blurred image later on. So you got a double advantage in the detectors, better DQE and the ability to sort of de-blur them later. And so that led to lots of really good structures, which is now how the power of the method has really taken over in structural biology. So I thought what I'd do now is show you 2 or 3 of the structures that I think show the method at its very best. And these 3 that I've picked as examples cannot be done by any other method than the single particle cryo-EM at the moment. So the first one is one of the ribosome projects. And obviously there were crystal structures of ribosomes that Tom Stites, Ada Yonath, and Venki Ramakrishnan were awarded a Nobel Prize for, all by X-ray crystallography. But in Venki Ramakrishnan's group here, the project that they were focused on after working on the lots of structures of different ribosomes was this mitochondrial ribosome project. And obviously, you have cytoplasmic ribosomes, mitochondria have ribosomes, chloroplasts have ribosomes, and so on. And of course, if you want to know the structure, you have to purify them. Alexei Amunts, who's now leading a group in Stockholm doing cryo-EM, he was a postdoc with Venki Ramakrishnan about 4 or 5 years ago. He had spent 2 or 3 years purifying this preparation of mitochondrial ribosomes, never getting it completely pure, the goal originally being to crystallize it. But then he got together with another postdoc, Xiaochen Bai, who's now in Dallas, who was with Shor Sherez, who'd written this new software called Rely On at that time. And they took images like this, which are impure, not very large quantities of this mitochondrial ribosome, beef heart mitochondria. And they got this, from images like this, they got this initial model from all the particles. But one of the features in Rely On and the other single particle software that you have now is you can ask them to look for different classes of structure. And in this case, Alexei and Xiaochen asked them for 6 to sort all the particles into 6 structures. And 4 of them turned out to be recognizable because of the size and comparisons to previous structures to being the mitochondrial ribosome, some of them being intact, large and small subunit, but a couple of the classes just being mainly the large subunit. And then there was another class that was the cytoplasmic ribosomes and others that they didn't know quite what it was. But by picking just the ones that were then the real correct structure they were after, and then using a second feature in Rely On, which is this idea of focused refinement, where you can find the orientations of the particles that you need to do to calculate the 3D structure. You can just pick the large subunit part, focus on that. And then if there is any flexibility, for example, the ratcheting that Joachim Frank discovered in 2000, then you get a really nice, accurate picture of the part of the structure that you focus on in the orientation determination. And then you can then subsequently focus on the other part, the small subunit, and get a sharp image of that. And by doing that, which is now the relatively recommended and standard approach in the 3D cryo single particle work, they were able to get a really nice model of all parts of the mitochondrial ribosome, small and large subunit, and build in lots of the polypeptides, many of which hadn't been seen before in any other structure. And then here is a little bit of their map. Obviously, they picked the best part to show. It's a little bit of Watson-Crick RNA double helix with 3 Watson-Crick, 4 Watson-Crick base pairs, and then a top view of one of them, where these would be the 3 hydrogen bonds in the Watson, and then an alpha helix and 2 loops of beta structure, including one that density they thought might be a magnesium ion, similar to a water molecule. So really nice structure from an impure preparation that was non-homogeneous and heterogeneous in structure as well, but still they were able to extract a really good. So this is a really good example of the different aspects from the preparing of the grid to getting the images to sorting them all out into different groups and classes. And a second one is the work on splicing and spliceosomes that was being done by Kiyoshi Nagai's group. And Kiyoshi had worked on this since about 1990, initially beginning with just 1 protein subunit that was very small, and then 2, and then 6, and then 8, gradually building up, all by X-ray crystallography. But he had a student, Kelly Nguyen, who had done her PhD by making crystals at something like 5 angstroms resolution of this tri-SNRP with 3 RNAs and a large number of proteins. And she was the one who led Kiyoshi's group. And unfortunately, Kiyoshi died about a year ago from liver cancer, but he did live to see the structure of very, very many of the spliceosome structures, intermediates, as it goes and does its splicing reaction. But this was the first one from their group, which was the tri-SNRP at 3.7 angstrom resolution. So that's like 2 angstroms better than they'd got by X-ray, and with many more subunits in it. And again, using this focused refinement, not all parts of it are as well ordered as other parts, but they were able to build a model of all of the subunits that you see there, the RNAs and the protein. And here would be a small part of their structure done 2 or 3 years ago now, including at the top a little bit of Watson-Crick RNA double helix, a little bit of single-stranded RNA, and then a stretch of one of the polypeptides, PRP3. So again, very flexible, much more flexible than the ribosome. And now they have 6 or 10 different structures of different intermediates that have been trapped using clever molecular biology tricks to arrest it, so they get a large proportion of the single particles in the right conformation. And then the third one that I'd like to show you is, this is an image of a membrane protein called mitochondrial complex 1. It's one of the biggest membrane protein complexes, 45 different polypeptides. John Walker in LMB in the 80s had decided that they ought to look at this and had done a lot of work on the molecular biology. But in the unit he became head of, there were a couple of other groups, Leo Sasanoff and Judy Hurst, who had focused on this mitochondrial complex 1, trying to get a 3D structure. And Leo had succeeded with a thermophilic bacterial version of it. But this one was beef heart mitochondrial complex 1. It's about 1 megadalton in size. It's about twice the size of homologous complex 1s from thermophilic bacteria. And Judy brought this sample over to our lab about 2014 or so, and Vinod Kumar took pictures of it, and within about a few months, 2 or 3 months, they had this complete model of the entire structure, showing at high contour level these 8 iron sulfur clusters, and at lower contour level 76 transmembrane helices. Obviously, they wouldn't have been able to interpret this without the help of the previous X-ray work on the bacterial homologue, but there were only 16 polypeptide chains, the core, about half the molecular weight in the bacterial, whereas 45 in this. But they were able to build the beginning of a model. But before I show you that, you can also go to a lower contour level and see the belt of the detergent and lipid that have been used to solubilize these molecules, pulling them out of the mitochondrial inner membrane. And then with about another year of work, they were able to get a 4 angstrom map, rather, in which they could recognize not only the core subunits, which are the blue ones that had a homologous previous structure from Leo Sasanoff's work, but, and these are the iron sulfur complexes built in now as an atomic model, but they could also see enough detail in the polypeptide for these supernumerary subunits, which are 29 additional subunits, such that they could uniquely identify all of them. And so now they have a complete model, 45 different polypeptide chains in a very big complex, again, with flexibility, and hasn't been done by any other method. But now Leo Sasanoff is now in Vienna and has done excellent work on the sheep version of the mitochondrial complex 1. Judy is now the director of the MRC mitochondrial biology unit, taking over from John Walker. And there are other groups in Germany and China who've done really good work. So this is now a really dynamic area, actually. But it's a third really good example of what the Cryo-EM methodology has done to help. And so I'd like to show 2 other structures that are not so dependent on Cryo-EM. Both of them had been solved by X-ray crystallography before, but they show you a little bit about how broad the range of applications that Cryo-EM can be done. And the first one is from the work of Miriam Koshai, who was another. There's a lot of students involved here because they see the way the wind is blowing and they act first, often before their leaders. So Miriam Koshai was a student with Radostin Danev in Wolfgang Baumeister's department at Max Planck and Martinsried. And she thought they ought to try and take images of hemoglobin. This is the one that Max Brutz had worked on. Human hemoglobin, this was her images. They were helped at the beginning by having Radostin Danev's faceplate that he'd worked on in Japan and developed over many years. And finally, they got one that was reasonably robust. And I think without it, they probably wouldn't have tried it. But now, actually, people are managing to do quite well without the faceplate. But anyway, by picking the particles, they got these 2D classes. they got a 3D structure, 3.5 angstrom resolution, the heme group, proximal and distal histidine, alpha helices, and a complete 3D model. So that's a 64 kilodalton protein structure that is probably at the lower end of what's able to be done by Cryo-EM at the moment. And obviously, you can go smaller, but the signal that you get that you need from each one of these particles, you can see they get, they're quite small, they don't have a lot of information in them, and you need enough information to find the position and the orientation of each of the particles so that you can average them into the 3D structure. And then the last one I'd like to show you is from a pharmaceutical company, and obviously they were always interested in how to determine structures so they could do better structure-based drug design. And this comes from a company called Astex, which is on the Cambridge Science Park. And they used to visit and collaborate doing X-ray work. But about 4 years ago, Haran Jyoti, who's CEO of Astex, said they'd been following the Cryo-EM and they thought maybe the companies should buy their own microscope. So actually, they went ahead and 5 companies, all relatively near to Cambridge, bought one of these Krios microscopes and installed it in the university in a nanoscience lab with it. They happened to have an empty lab that had a Titan for material science that had been removed. So there was a nice empty lab. And so within about 6 months of having the idea, they had 5 companies paying for 1 day a week each, with the university and LMB having some of the weekend time that was available. And this is one of their first papers. This is Pamela Williams and the people at Astex who had decided to pick beta-galactosidase, which is an enzyme from E. coli, comes off the lac-operon, LAC-Z, and which Sriram Subramaniam actually had worked on while he was on his sabbatical here, and had a 1.8 angstrom resolution structure on it. But their idea was, could they successfully get routinely and daily maps good enough to do structure-based drug design? And so they worked on it for probably about a year. They collected, say, something like 60 data sets in complex with lots of different ligands. And so here's the structure. And nearly all of them are one day data collection, and 2.3, 2.4 angstrom resolution structures. Here are 1 to 3 different ligands in which they're able to recognize the shape of the sugar and the conformation of the hydroxyls and so on. And then at the bottom, they've added in another, which is one of their cancer drug targets, a pyruvate kinase structure with 2 ligands that they were actually more seriously interested in. So across the range of structures, both those that could be done by X-ray crystallography and those that couldn't because they're too big, too flexible, too impure, there's been a lot of impact. So what about the field as a whole? And one of the things that we've always done is to try and follow the progress of the different methodologies in terms of depositions in the protein data bank. And back in 20 years ago, there was nothing really from electron cryomicroscopy. Even though you could get structures, they weren't good enough to get atomic resolution. And really, the first ones were the icosahedral viruses with the new microscopes, but before the new detectors came in. So all the ones that are, say, between 1999 and 2010, most of these depositions would be icosahedral viruses or filamentous structures. But you can see X-ray crystallography still continues to grow, 10,000 PDB depositions per year. NMR peaked in 2007 in terms of de novo structure determination. It's still very good for weak ligand binding, substrate binding analysis, but de novo has dropped off, whereas the Cryo-EM didn't really start to be visible on the same scale until a few years ago. So the one plot that we like to look at is the ratio of PDB depositions done by X-ray compared to those done by electron microscopy. And you can see before the year 2000, for every for every one cryo-EM structure that had an atomic model being deposited, there were over 1,000 or 2,000 X-ray structures. So it was just nowhere to be seen. And then these new microscopes with better field emission guns, much better, higher vacuums, more stable stages, they came in around about the year 2000, the Polaras and then the Krioses. And so then it was more productive, so it dropped to about 100 to 1. And then the new detectors came in around about 2012, 2013. And then it started to, as the people who are already doing it started to make use of it, and then other people from other methods saw how productive the method was and started to use the methodology as well. And since then, it has grown exponentially. So this is from 100 to 1, it's down to about 4.5 to 1 now. And these last 2 points, the second last one is the ratio for last year, and this one is for the first 3 months for this year. So if you then try to extrapolate that, assuming it'll carry on, of course it could saturate, then it turns out you reach sort of parity in about 3 or 4 years' time, mid-2024. And so that does depend, though, on continual investment in microscopes, and I'll come back to that at the end. How can we get enough capacity to satisfy the need for demand and for productivity? But before I come to that a little bit, I just show 2 other structures that I think I really like them because they illustrate aspects of the method that look forward, and they're both to do with what about higher resolution. So one paper that I really like comes from Dimitri Lyumkis' work at the Scripps, and he took note of all the recommendations, all the tricks, all the best practice that other people had recommended, and worked on this adeno-associated virus, which, and here's their structure. It was at 1.9 angstrom resolution. But the reason I like this paper, it was the first time that in cryo-EM, the use of an eVolt sphere correction or an eVolt sphere mathematics in the algorithms for making the images, rather than just assuming this image is a projection of the structure. It does an X-ray with 1.5 angstrom, 1 angstrom has a very highly curved eVolt sphere, but X-ray, because the wavelength is so short, it's almost flat. So you have to get to really high resolution on bigger structures. And they found that the resolution went from 1.9 to 1.8 roughly by including the correction, which was done already by Matthias Wolf and Nico Grigoriev inside their FreeAlign software back in 2006, but it had never been shown to work until 2 years ago. And what they found is when they reprocessed the data with the switching on of the eVolt sphere correction, the blurring factor, that remember it was 3,000 back in the adenovirus days in the 80s, dropped from 60 to 50 square angstroms. And that's roughly 40, 50, where all the best structures are nowadays. And of course, when you then look at features in the structure, all of the side chains look really nice. And then the other structure, which I should mention, is work that many groups, several groups, have done on apoferritin, because it's got 24 copies in each particle. So in an image like this, where you might have 100 or 200 particles in one image, you actually have several thousand asymmetric units. And so from one image, you can get quite a good high resolution map. But of course they didn't. They took lots of images and they got a 1.22 angstrom resolution map. I haven't shown any data, but that was work done by Takanori Nakani in Shor Sherry's group in collaboration with Thermo Fisher, with all their latest gadgets, better detector, better energy filter, cold field emitter, and so on. But other groups have done more or less the same, actually, which is Holger Stark, for example, and Wachu at Stanford, without all the sort of fancy things. So eventually, you're going to be able, because if you take pictures of gold in a Krios, you can get way beyond 1 angstrom resolution. So there really isn't now, if you're careful enough, any particular reason why you don't go to higher resolution. But okay, so show two other slides after this. I'm not going to say anything about it. I don't personally do tomography, but those of you who are interested in cryo-EM must know that electron cryotomography does have a great future. It just hasn't had quite the improvements that you've seen from the single particle cryo-EM from the new detectors. It has an improvement, but this picture shows how electron cryotomography was about 20 years ago. It's from Ohad Madalia's work with Wolfgang Baumeister at Martinsried, and it shows they were able to find membranes, which are blue, actin filaments, which are in brown, and then other macromolecules, mainly ribosomes, big assemblies, which were in green. But if you then fast forward from this to a year or so ago, from the work that was done by Julia Mahamid, electron tomography of HeLa cells, now the quality from the new detectors, the movies, the better field emission and so on, is much higher than it was back then. And so now in these segmented tomograms, you still see the membranes and the actin filaments and so on. But if you look at the ribosomes now, they can identify the part of the blob that is the large subunit, the small subunit. They can see which ones are ratcheting, which ones are bound to the translocons in the membrane. So much more detailed. And then in the membrane, these are the nuclear pores. And by extracting these particles that you see in a single tomogram and doing subtomogram averaging, you can get higher and higher resolution. And there's no reason in theory, as long as you can find the particles, extract them and average them, why you shouldn't do just as well in tomography as you do in the single particle work. Okay, so that's basically a little bit about the history, the overview, where we are now. So now I'd just like to say a few things about what are some of the outstanding problems and some of the things we're interested in Cambridge. So this is, we debate this all the time. The method works. Many people say they are incredibly happy, they don't need any changes, but we know that it's not as good as it should be. And so there are many things that can be improved with the hardware, the software, but particularly with the way you make specimens to take images. So one of the things that we work a lot on, we have about 4 or 5 projects ongoing still for detector development, because that's still a sort of rate limiting step. And you can always have bigger, better, and faster detectors. There's no limit to how far you can go. And you've seen this with digital cameras, how every year there are better ones. And unfortunately, because of the way the electrons react when they hit a solid surface of the detector, you do need a different design, not entirely different, but somewhat different, for 300 kV and for 100 kV. And I'll say why we're interested in that in a little while. But that's one way forward. And then this middle part of the slide is the thing that many of us worry about a lot, and that is, why do so many cryo-EM structures that people publish nowadays start with millions of particles? And I've seen them with people take 50,000 images, and then they extract millions and millions of particles, and then they do this classification that I've talked about before, and they gradually get it down, and then they end up with a nice structure that they're happy with, but it only has, say, 50,000 particles in it. So they're down to 1% of what they started with, or less than 1 particle per image sometimes. So obviously you can rationalize this, but I think there are lots of things that are not understood at the moment. And many people are quite interested in what is the air-water interface doing? Because it's been known for decades that all proteins, of course, tougher ones take longer, but they denature at the air-water interface. So they unfold, or multi-subunit ones can disintegrate. That could be part of the reason. Many people notice you get preferential orientation. If you only have one view of a particle, you can't get a 3D structure. So people do tilting tricks to get around it. And then, like in the mitochondrial ribosome, you get mixed states, different conformations. These are all reasonable, but there is this possibility, and I think Ed and I had a little chat earlier about how the stresses and strains that are introduced into your specimen when you do this blotting and plunge freezing method might not only stretch and bend filaments, but individual protein subunits. And that's not an area that's been investigated very well. And of course, one idea is that perhaps you should treat EM grid preparation just like affinity chromatography. You make a really nice mild column of a material that is very bland and neutral, has no effect on the protein molecule. So you make a grid surface, you passivate it with polyethylene glycol, and you make, say, histidine, polyhistidine ligands, ligand binding areas, nickel NDA or something. So you want an affinity grid so you can attach your protein to it. You don't need to concentrate it up to 5 or 10 mg per mil. You can use it at much lower concentration. So these are all areas that are unsolved, and we worry about this all the time. And the big thing that I find most discouraging and annoying is that if we have an idea, we cannot get on the microscopes because so many people are using, and we have about 10 or 11 cryo-EMs in the lab, 3 Krioses and so on. You can't get on them because many people have got good specimens, they just want to collect data. So if you want to explore all the problems, so we were driven to the idea that really we need to have some way of going forward where you can get more microscopes that are not as expensive. And one of the things that we did, this is Vinod Kumar and I, when we wrote a review, that was after Vinod had been doing this mitochondrial complex 1 structure, and before he went back to Bangalore in India to help to set up the Indian Cryo-EM facility in Bangalore. We wrote this review in Quarterly Reviews of Biophysics, and we put this little section in about how we should be planning to go ahead so that we don't run out of exponential growth, so that we can do debugging of specimen preparations, or if you just want the structure, you try all the different tricks that are needed to get a really good specimen so you can get data and get your structure that you need. And so what we said is that obviously one way is to have big national or international facilities, and we have in the UK, we have EBIC at the Diamond Synchrotron. They have about 7 Krioses. In the US, there are 3 national facilities with similar numbers that are available for everybody. But before you go there, you have to get your specimen to work. And to do that, you need to get, we think, preferably daily or maybe 5 times a day, so that you could, within one day, you could go from not having a good specimen to having a good one, rather than having to wait weeks or months in order to debug them at the moment. And you also need the microscope not just to be a cheap one that works like they did 20 years ago. It needs to be one that has a bright source and a good detector. And so what Vinod and I said, we desperately need an inexpensive microscope. And our suggestion was that you should go for 100 kV with a field emission gun, and there was none available at the time unless you bought an expensive one, in which case you can use the existing detectors. That was our idea. And we tried persuading the companies to do this. And obviously they were still being stressed trying to deliver enough of the high-end microscopes. So they didn't give it a very high priority. But now I think they're both beginning to, well, all of them are beginning to think about it. But as we went along that route, before advocating it too strongly, we thought we needed to do 1 or 2 experiments. And I'm just going to show you those at the moment now. So the first one is from work that's done in collaboration with Chris Russo's group. Matthew Peat is a postdoc with Chris Russo. And so what we thought we should do is to look at all the older data from Bloch and Better, who are Nobel Prize winners in physics, but they also wrote a lot of papers about electron scattering by matter. So elastic scattering and inelastic scattering. And inelastic scattering deposits energy, and part of that energy does radiation damage, breaks bond, ionizes atoms, and so on. And these 2 curves, labeled sigma I and sigma E, are the elastic and inelastic scattering cross sections from the Bloch and Bethe theories. And you can see these lines on on a log scale versus electron energy, they're not exactly parallel. In fact, they get closer together as you go to lower energy, and that means there's less inelastic scatter for the same amount of elastic scattering. And it's elastic scattering that gives you these beautiful high resolution images, and the inelastic scattering that does the damage. And so we thought we should remeasure these, and that was a lot of the work Matthew did. The full lines were experimental remeasurements of these theoretical curves, and they fit pretty much. And the radiation damage parallels pretty much the inelastic as well. So it turns out the bottom line is that not only would a 100 kV microscope with a field emission gun and a new detector be cheaper, but it would also give you more information for the same amount of radiation damage. So it should be better. And so that was published about 18 months ago. We thought we ought to actually just show by taking data that this was also practically true. And so there's another paper that's by Katerina Najdanova et al., published about sort of 9 months ago or so. We looked at 4 or 5 different structures, but this was the one deep and only done over a few days by converting a 200 kV microscope with a field emission gun, turning it down to 100 kV, and putting on a Dectris detector that was designed for X-ray work, but also happened to work well at 100 kV. And now they are marketing that. They have a singler that you can buy to put on 100 kV detector. But we got a 3.5 angstrom structure with a few hundred images with a detector that's 500 pixels by 1,000 pixels. So it's 32 times smaller than the detectors that you get for the 300 kV microscopes. And so we said now, in theory, by experimental measurement of elastic and inelastic cross sections, and by experimental measurement of some macromolecular structures, it does look like Cryo-EM at a lower voltage is viable. So then I thought, and some of you will have seen this, I thought I'd just end with 2 slides. One of them is from a webcast that Thermo Fisher presented in November. So they have launched a product which, as you can see, exists at the moment only in the computer. So it's a video presentation, but this is the loading bay. One grid is in this holder and it goes into this hole, and then you're off and running with a diagnostic microscope, which, because they launched it, you can actually order it now, and that will be less expensive, but we'll have to wait to see how good it is. They haven't got a very good detector in it at the moment. And then the other thing we did was that we wrote a grant about 2 or 3 years ago, together with Jim Naismith at the new Rosalind Franklin Institute on the Harwell campus, and Dave Stewart, who is involved with Diamond, and got money from the Wellcome Trust to go forward with a sort of lab-based prototype demonstration project. And this is one of the microscopes that we've bought. That's a JL 1400 that costs probably about $250,000, something like that, maybe $300,000. And on it, it's got a Dectris detector. It doesn't yet have the field emission gun, but there's a company in York in the UK, a small company, developing a field emission gun that's going to go on this in a month or 2. And then we've also bought 3 of the existing cold stages from Catan, the ELSA, really beautifully engineered now, Fiscioni, and Simple Origin. And so our idea is that, let's say later this year, there will be a couple of, Thermo Fisher hopefully will have their one, we will also have this one, and we're hoping that both of the companies will go forward and make Cryo-EM not only as productive as it is already, but also able to be scaled up with less expensive instruments that are actually as good as, but maybe in the beginning are a little bit more hands-on rather than computer controlled, and then take us into a sort of a new era of Cryo-EM so that we can continue being productive and solving all the problems that need to be solved to make progress. Okay, so I'm going to end there with just some of the acknowledgements. Of course, I've shown mainly the work of other people or other groups. some of them from the LMB, adenovirus from Jacques Dubochet, Phoebe Stewart, and Hong Cho's work, the mitochondrial ribosome from Alexei Emuntz, who's now in Sweden, Xiaochen Bai, now in Dallas, the spliceosome, Kelly Nguyen, who worked with Kiyoshi, did a postdoc with Eva Nogales and is now back here as a group leader at the LMB. Vinod Kumar is in Bangalore. Shai-Pai Ju is back in China. Judy Hurst, now the director of her MRC unit. Miriam Koshai, who's the one who did hemoglobin with the faceplate, works for Novartis in Switzerland now. The apoferritin, the beta-galactosidase is two examples of looking forward. And then Dimitri Lyumkis and Shores with other, let's say, cutting-edge, high-resolution methods. And then obviously cryotomography is another, not completely separate. You need the same equipment, but it needs a different set of skills to make good progress with that. And then we still continue with our work on both detector developments with Greg McMillan, Wazi Faruqi, and the two detector people. And then Chris Russo's group have taken over this 100 kV project for development. So hopefully that's given you some, let's say, positive view about how better than we thought it would, the method has developed and how much progress there's still available to go forward in the future. So many thanks.
Well, thank you, Richard. That was wonderful. Let me just start off with the first question. Crystallographers have pointed out to me that the software in crystallography is so mature now and so good that most people solving crystal structures really have no great expertise in crystallography. It appears Cryo-EM is still far from that. And maybe you'd want to say something about the level of expertise still required.
Yeah, I'm not so sure that that's true, actually, because, you know, in our lab in Cambridge here, before the new detectors came, we had probably about 12 or 15 people doing Cryo-EM. We could walk up to we had, 4 or 5, we could walk up to the microscope every day and get on it. There was no one competing for time. But when the detectors came, and then we got a Krios, and we had the first Krios in Britain, and the number of people using it at the very beginning weren't so many. We opened it up a third of the time for people in the UK. People were not ready with specimens, but as they knew what was needed, it grew. And we now have something like 150 or 180 people in one little lab out of 400 or 500 scientists. Maybe a third of them now are doing Cryo-EM. They don't do it full time, and they certainly don't know what's involved. They are using it just like the X-ray crystallographers use it. Now, you say we're unusual. There's a lot of infrastructure. They don't need to set it up for themselves. They can just ask someone. And we've had lots of, often it's the student, actually. The one that I remember was Rebecca Voorhees, who's now group leader, assistant professor at Caltech. She was another student with Venki Ramakrishnan. She did her PhD doing Cryo-EM, doing X-ray crystallography. Then she went with a cell biologist who had no Cryo-EM experience and decided to work on ribosome translocon complexes. This is about 3 or 4 years ago now. And she knew zero about Cryo-EM. But she knew what she wanted to do, and she was quite persuasive in terms of getting access to people. So she began by going to the slaughterhouse to get pig pancreas and then extracted and purified ribosome translocons. That took her about 2 months. Then she said, okay, I'm ready for EM. And they said, okay, negative stain. So she did negative stain. That took about 2 weeks. She said, okay, now I'm ready for cryo. They said, okay, well, pilot project, a little bit of cryo, 2 weeks, worked out quite well. Then she was ready for the big time. And within about 3 months or 4 months, she had a structure. And the paper came out, Voorhees et al. In April, the beginning of April. 6 months from knowing nothing to having a first author paper in a journal. And I think that's happening a lot. So I think that's one way things will go, there will be 95% of the people won't want to know. They just want to use it as a method. However, as I was trying to point out, there are lots of problems. So you need to have other people who are interested in solving the problems and making it more user-friendly. But I think it's going to continue on this exponential growth, particularly if we can get some cheaper microscopes so that, let's say, every group could have their own microscope. You can get on every day. You might even be able to solve structures without going to the national centers. So I'm relatively optimistic that we've already gone quite a long way down that road.
Good. So this is open for questions. Just use the chat function to indicate you have a question. In the meantime, let me just ask about ridiculous structures. That one thing that expertise provides is the ability to see that something is clearly wrong and makes no sense, and it's still possible people can produce garbage in Cryo-EM and not realize it.
Yeah. Yeah. Okay. So, I mean, many of us were very worried about this about 10 years ago, because people were publishing structures and it was known, I think everybody accepted it ought to work at atomic resolution. And so people were pushing and pushing it. And it wasn't realized that you could get, just like an X-ray actually, just like before we had Axel Brunger's method having R free and R work, people were overfitting X-ray data. And the most famous one, I think, is where one of David Eisenberg's structures had the Rubisco structure with the small subunit traced beautifully and correctly, but backwards, so that the amino terminus was the C terminus and so on. Okay, so we had the same thing with Cryo-EM, and I certainly worried about it because it had a reputational risk for the field, that let's say there'd be some big scandal, like there was with some of the early, for example, metallothionein was done correctly by NMR, but wrongly by X-ray, and that put a cloud over the whole field. So we did have various things like, for example, our particular one was tilt pair validation, where you take two images of the structure at different angles, and then don't let the computer know that those are related, but you know that they're 10 degrees related, and then you can check up on that. And that was very popular for a short while. But now that the detectors have become better and the structures have become better, you can often just look at the structure and see if you can see beta sheets resolved 4.8 angstroms apart, or if you can see alpha helices with the pitch and side chains coming out, you can tell, have you got the right hand? You can tell that you've got it. So actually the use of these validations has kind of dropped out now, but they're still available. And then the second one we did was, we called it high resolution noise substitution. So you take all your images, either the original micrographs or the single particles, you Fourier transform them, and then beyond a certain resolution, let's say you pick beyond 4 angstroms, you simply randomize the phases. You leave the power the same, but you've put in nothing beyond that resolution. And then you just do exactly the same computing, and then you look at the resolution plot, the Fourier shell correlation plot, and it should drop to 0 at the resolution where you randomized it. And if it doesn't, you've got overfitting, regardless of the software that you're using. And again, so that has some of the programs like Reliant, they incorporate that at their final step of resolution estimation. And then if you've done it, if you put a mask in that creates an artificial, for example, it gives you a warning. So there are sort of hidden built-in validations at the moment. And we haven't really had any, well, we did have the HIV envelope glycoprotein. The people who did that, they were simply deluded. They'd built an atomic model and they thought it was high resolution, and it wasn't. It was low resolution. So I don't think that there's not such a serious reputational risk to the method now. but there, of course, there would be individual risks. But now I think the refereeing and the awareness in the field is such that it's not, there's still, I would say, a little bit of, let's say, resolution estimation over optimism. So, because if you use all the little tricks that are in the programs now, it's not so clear where you've introduced the bias. But fortunately, it only gives you it only seems to exaggerate the resolution slightly. So it's reasonably safe now, I think, with that being going forward. So I'm also reasonably happy about that.
Very good. Okay, then the next question is from Mark Yeager. Mark's unmuted.
Yeah.
Beautiful presentation, Richard. I was wondering if you could comment on the advantage of electron microscopy yielding Coulomb potential maps, and thereby the electrostatic interactions within a macromolecule versus electron density maps provided by X-ray crystallography.
Yeah. Funnily enough, I was just reading a paper yesterday by one Mark Yeager.
It's a close relative.
So indeed, it's true that the scattering factors for atoms, scattering factors for electrons by atoms are not very well worked out at the moment. And indeed, if you have let's say, a negatively charged oxygen or a positively charged nitrogen, that charge will have a big effect on the scattering of the electron, whereas it has very little effect on X-rays. So for X-ray scattering cross form factors, you can simply count the number of electrons. It might have a slightly different Gaussian, but say carbon, nitrogen, oxygen, they scatter as 6, 7, and 8. But if you look into the international tables for crystallography at the scattering factors for carbon, nitrogen, and oxygen by electrons, the tabulations are for non-bonded atoms. And in that, at resolutions that we're typically working, say 3, 4, 5 angstrom resolution, instead of carbon, nitrogen, oxygen being 6, 7, 8, it's more like 8, 7, 6. And that's because in unbonded atoms, the shells are not properly paired. But as soon as you form molecules, the orbitals shrink. And what we haven't got is properly published, and certainly not in the programs, the scattering factors for the bonded atoms at the moment. And actually, Nico Grigoriev and I, we tried to refine those numbers experimentally in 1996. And we did get something like, we knocked 0.5% off the R factor for Bacteroidopsin by properly modeling it. But the data weren't so good, and that hasn't been carried on. And I know Jim-In Wong at Yale with Peter Moore, and Mark will know about those as well. He's very keen to try to use the fact that the maps are different to make intelligent deductions about the nature of the structure and the charges. The problem is, at the moment, you've still got radiation damage. And often the radiation damages the side chains that are exactly the ones you want to know about the charge on. So aspartic acid and glutamic acids, and they fall off. It was known by X-ray crystallography that those were the residues that were damaged, say, in lysozyme, the active site glutamic and aspartics get damaged before many of the others, and then phenylalanine, tyrosine, tryptophan are much later. But as in X-ray, I think one idea is that if you've got a series of observations like you do with the movie that you get when you record X-ray images, you ought to be able to extrapolate back to 0 dose and get both the average structure, but also you should be able to get the undamaged structure, perhaps not quite so accurately, and then you could perhaps compare them. And I know that Chris Russo recently, with his gold grids, and then some of the improvements in particle polishing in the software for doing 3D EM, they are getting better at extracting the information from the first few frames, which could be the ones that are needed to give you that. So yeah, so I don't think at the moment, I wouldn't believe any of them at the moment. And I know that Yoshi Fujiyoshi published papers about bacteria redoxin back in the 1997, 1998, where they tried to say that the differences in the density of the aspartic acids were due to the ones that weren't there were negatively charged, and the ones that were there were positive. But actually, the ones that weren't there had been radiation damaged and removed. And so, but looking forward, this is something to look forward to. You should be able to use exactly that information, but until someone does it properly and then publishes the form factors for neutral atoms as well, or atoms, for example, methyl groups, CH3 and valine or isoleucine, CH2, CH, CO, carbon atoms in benzene rings, they should all have slightly different scattering factors. And hydrogen scatters more for electrons than it does for X-rays. So this is all something to look forward to. So one of the projects we would like to do is to do one really good structure at highest resolution with lots of data, so there's no noise in it, and extrapolate it back to zero, preferably a structure with some phosphates, sulfates, iron atoms, and zinc atoms in it, and then ask the theoreticians. And here we have Garib Mushadov, who's a group leader here at LMB, who's been involved. He writes RefMac, and then there's the Phoenix software. What we want is a little button you press. You say, I want X-ray, I want electron form factors, including charges, rather than X-ray form factors. But it won't make a lot of difference, because once you've got a side chain, that side chain is there, whether it's got X-ray or EM form factors. So it'll make a small difference, but it could be that some mechanistic feature of a particular structure would be helped greatly by knowing about that. So I think that's another area to be positive and optimistic about.
Okay, the next question is from Meg Sales.
Yeah, so it's about eVolt sphere in real space. eVolt sphere is indeed in reciprocal space. So obviously the way it works is you have an image. You have an image of a single particle, and it has Fourier components. And if this is your particle, you irradiate it, it diffracts, and then you focus it back. And if you don't, if you have it nearly in focus, then the Fourier component from the diffraction to the right and from the diffraction to the left will end up being overlapped, and then you can't disentangle them. But Matthias Wolf and Nico Grigoriev and actually David Rosier, they have a paper in 2006, which was programmed into FreeAlign 14 years ago, that allows you to switch on an eVolt sphere correction. And then actually a little bit later on, Chris Russo and I did some work with gold diffraction. And then we said that in addition to the idea that Nico advocated in 2000, you should also try and use a defocus that was big enough to keep the Fourier components. In other words, you've got the curved eVolt sphere, the two Fourier components come out at different angles. You should defocus it by enough that those two Fourier components are resolved in the image. And then you can mask them out, and you don't have them overlapped and don't have them confused. And then so that's now been shown for quite a lot of structures, particularly the ones that are higher resolution or bigger. So viruses at 3 angstroms, or smaller structures at, say, 2 angstrom resolution. So Relion and System both include this now as a correction. And the correction is always done in reciprocal space from the image, but you take part of the image, the real space image, has the Fourier component, let's say, that's deflected to the right, separated from the Fourier component that's deflected to the left, because it's been defocused by, say, 1 micron or 2 microns. So in the image, the two eVolt sphere samplings that are in different parts of reciprocal space, they're recorded in the image, not overlapping. And then you can extract that information and put them into the 3D structure correctly. And that's what gave Dimitri Lyumkis and then all the other people who've now used this the way of getting slightly higher resolution by doing the EVOL sphere correction, which is sort of a mixture of real and reciprocal space.
Okay, next
Okay,
question.
thank you.
Sure. Michael Wiener on micro ED.
Yeah, hi, Richard. Yeah, I was just curious. I mean, another advance that appeared in the last decade, of course, was by my reading, Tamir Gonin, building on previous results by, to my reading, primarily David Stokes. Solving the multiple scattering problem and developing microelectron diffraction, and then of course Andrew Leslie contributed some outstanding software. So I'm curious of where, if any, do you see a role for micro ED? I mean, my first thought is methods used to make very small crystals, or potential convergence between the kinds of sample needs for free electron lasers, maybe smaller macromolecules, there could be a role, but you still need a crystal. And is that really, is there going to be classes of problems where you're really going to have to go to the trouble of coaxing a 3D lattice?
Yeah, I think we should encourage Tamir Gonin and the micro ED community to do the very best they can, but I don't have the same sort of deep-seated positive feeling about it. So one is you do have to make crystals, and part of the reason that my own interest in our own little group switched from, we tried very hard to make 2D crystals of lots of different membrane proteins, and it turned out to be just as hard as making 3D crystals. And then we started, because of the difficulty making 3D crystals, say Chris Tate and we got involved in making the proteins more stable so that you could get crystals where you couldn't do with the native protein. And so there is a way forward there. But at the moment, saw the hemoglobin structure with improvements in the grids, in the way you make the grids, and in the detectors and the microscopes. I think that the size minimum, where you can't go beyond, it's around 40 or 50 kilodaltons now, you can get a structure with, and the smaller you go, the more work it is, because the orientation is not so well defined, and then you get a bigger B factor, and you need more data to make up for the loss of signal. But I think there's a great future in bringing the size limit down. And then, of course, there are people who are quite keen on making adducts. So let's say FV antibody complexes to some smaller protein. So that's where I think that's you're opening up a gate into a green field with a great future. Now, the thing about the micro ED is, of course, you can do things that are small. Once you're down to about 7 or 8 kilodaltons, and if you dissolve your polypeptide, let's say it's 30 amino acids, that might be 4 or 5 kilodaltons, and you take a picture of that, you really will find it hard to tell the difference between just water and water with your 4 or 5 kilo, because the noise level that you're allowed, is about the same. So there will be a size limit. So potentially you can make a 3D crystal, and then you might be able to do XFEL and synchrotrons as well. So there will be, I would say, there'll be a small niche. Now, it used to be, I remember there was a Gordon conference actually, and it was always felt X-ray and NMR were the powerful methods, and then Cryo-EM was the niche. Whereas now, the sort of tables have turned a little bit. But I think the micro ED will remain a useful niche. And for example, I think a lot of the medicinal chemists synthesize a compound. They think it's the drug that they want, but they don't know for sure. They need an X-ray structure, but it's only a 500 or 1,000 molecular weight small molecule. And so, and I think Tamir's realized that actually, and I think a lot of his emphasis has switched from membrane proteins, calcium ATPase, protease, to smaller structures. And obviously the manufacturers, they love micro ED because when you buy equipment, it doesn't matter whether you're doing tomography or cryo-EM or micro-ED. So they like to sell it to you. So, but you have to, as the customer, as the user, you have to, as you do when you buy a new car, you have to see through the advertising and sales pitch and make your own decision as to whether, you know, this is a big thing or is it a minor thing. Yeah.
So in some sense, micro-ED as the next small molecule crystallography, in some sense, a routine analytical tool to check the identity of what you've synthesized.
Yeah, yeah. And I mean, obviously, once you've got the once you've invested in it, you know, you could, in principle, you could schedule Monday for micro-ED, Tuesday for tomography, and the rest of the week for cryo-EM, for single particle cryo-EM.
Thank you very much.
So I want to thank Richard as well. I need to close the questions now because Richard has a session with postdocs and graduate students in about 10 minutes, and I'm sure would appreciate a break. So thank you so much, and we look forward to your physical visit to UVA at some point once the pandemic is over. We'll be in touch.
Great, Ed.
Thanks again, everyone.